My name is Ruth Carmichael and I'm a Postdoctoral Research Fellow in the lab of Professor Michael Schrader at the University of Exeter. So in the Schrader lab we're interested in peroxisomes. These are small compartments that are found in pretty much every eukaryotic cell, and they're important for a range of metabolic functions, particularly to do with making and breaking down specific fat molecules. So they break down very long chain fatty acids from the diet to turn them into energy, and they also make a lot of important fat molecules, for example ones that insulate the neurons in the brain. As well as that, they're responsible for maintaining the levels of reactive oxygen species in the cell. They're very essential for human health. Patients with inherited peroxisomal disorders have severe neurological and developmental symptoms, but more broadly, peroxisome dysfunction is implicated in a number of other diseases like cancer, neurodegeneration and ageing. So we're particularly interested in peroxisomes in human cells, which very little is known about, and how they are regulated and how they function, so particularly how their number and size are controlled, how they respond to the environment and how they communicate with other compartments within the cells. And ultimately, by understanding this more on a fundamental level, we're hoping we can then exploit that for benefiting human health. In order to understand more about how certain important peroxisomal proteins are regulated in the cell, we need a method to measure their expression levels endogenously, and to do that we've adopted an endogenous tagging strategy where we've used CRISPR to introduce the NanoLuc sequence in frame with our genes of interest, so they're then expressed as fusion proteins. And we've also introduced firefly luciferase gene as well under a constitutive promoter using recombination, and this allows us to be able to account for any non-specific changes in our cells. So for this project, the NanoGlo DLR assay is completely essential because we need to measure both NanoLuc and firefly signal in the same cells, and it's very easily done with this assay. We also use some of the other assays, including the CellTiter-Glo for cell viability. We chose the NanoLuc for endogenously tagging our proteins because of its small size, its sensitivity and its amenability for high-throughput screening. We checked in advance and found out that tagging our proteins with that has no effect on their functional localization, which was essential. And then being able to combine measuring the NanoLuc signal with the firefly luciferase signal in a single assay saves us a step of then having to normalise for cell number and cell viability using a separate assay. The NanoGlo DLR assay is really simple. It only requires a couple of buffers, so as a result it's really quick to do, there's not a lot of wastage, and it always gives us really good reproducible results. The CellTiter-Glo is a great assay for cell viability. It is so much quicker than some of the more old-fashioned ways of doing it, like MTT assays, so that's been really great. For this project, we use the GloMax Discover plate reader, which allows us to measure luminescence, but we also use it in other assays in the lab for fluorescence and absorbance. It's ready optimised for all the Promega kits we'll be using, and particularly useful is the fact that the protocols for them are already loaded on to make it incredibly simple to use. And also we like the fact that it's upgradable, so we can add additional fluorescence filters or injectors as we need it in the future. There's so many automated features as well that you can just essentially program a protocol, leave it, and it will do all the steps from incubating, shaking, measuring itself. So it's really simple, and you know it's going to work first time. To be honest, I think the GloMax Discover is the best bit of equipment we've ever bought. It's ended up being far more useful than we anticipated. We don't only use it for the luciferase assays, we now do a lot of our routine protocols on it. Another reason that we picked the GloMax Discover in particular was the fact that it was versatile and would take a wide variety of different plates, so we can do everything from 6 well up to 384 well, which will be ultimately really useful for the high throughput screening. So far we've started to understand a bit more about how the peroxisomal proteins we're interested in are regulated. The next step is to look at how that affects peroxisome number and function. We've recently had a paper published in Journal of Cell Science looking at how peroxisomes proliferate, so how the number increases in the cell, and found an alternative pathway that hadn't been characterised before. And hopefully we'll be able to use the NanoLuc assay to then understand how this protein is regulated and how it might be affected by environmental conditions. So once we further optimise this assay, hopefully we can then take it into higher throughput screening in order to identify novel compounds that will regulate these peroxisomal proteins and then hopefully ultimately peroxisome functional number. So I really like using the Promega kits because we're getting some really nice data out of it that hopefully will one day be translational into more biomedical research. So it's really satisfying to know that we are actually producing something that will maybe help human health in the future.